A very good evening and welcome to another session of NEET Bytes by Meritnation in Biology. Hi Aditya, good evening. I hope you're doing great, dear. So, welcome to another session of NEET Bytes by Meritnation. Hi Sandhya, hi Meghna, good evening, everyone. So, I hope everyone is doing great. And today we will continue. Hi Lavida Bas. So, today we will continue with our same unit, and that is biology in human welfare, right? So that is the unit we are going to finish off at a stretch, and I'm going to take up your two chapters over there. One is strategies in food enhancement and microbes in human welfare. Fine. So strategies for enhancement in food production is one of the topics over there, and the second topic is, okay, Lavi has already answered the first question. Okay, what about others? Hi, Banu. Good evening. So, Aditya, I hope you are all set. Okay. So, homozygous pure lines in cattle can be obtained by the first option is mating of related individuals of same breed, mating of unrelated individuals, mating of individuals of different breeds, or Smriti has given me option B as the answer. Sudarshan says it is A. Amreen, good evening. Sandhya says it is A. Okay, Meghna says it is A. So, hello Shivam, good evening. So, very well. Well, all of you are right, and the correct answer is option A. Right, we know if we want to produce pure lines, and the word pure lines over here means homozygous, right? Whether it is homozygous dominant or homozygous recessive. Irrespective of that, if it is a homozygous strain or a homozygous individual for a particular trait, it is a pure line for the same. Right? That is the same definition which Mendel has used and will go by the same. And accordingly, mating of related individuals of the same breed will lead to accumulation of the same kind of alleles in the forthcoming generation, and thus formation of pure lines, right? So that is the answer over here. I hope it is pretty simple, very easy for all of us to understand. And let us see the next question of the day, right? Okay, and here comes the next one. A system of rotating crops with legume or grass pasture to improve soil structure and fertility is termed as what? So this is again a very easy one, and I'm expecting a very quick answer from all of you. Can it be contour farming? Can it be strip farming? Can it be shifting agriculture? Or is it lay farming? Come on, guys. Let's make it quick. Everyone seems to have gone absolutely silent at my end. I don't know why it is so. I hope the chat is working for all of us. Okay. Okay. Looks like it's all silent here. Okay, looks like the chat is working. I am not pretty sure whether it is working for everyone or not. Okay, looks like it's working. Anyways, over here, the correct answer for this question will not be the option A. Contour farming is farming basically on the slopes, right? And farming in the same pattern as is the pattern of the slope design. Strip farming, just cutting off something and farming over there is strip farming. Shifting agriculture, the agricultural land is being shifted every now and then, right? It is a kind of crop rotation, and such a kind of crop rotation where a legume or a grass pasture is grown during the off season is termed as lay farming, right? So it is lay farming over here. I hope this is clear to everyone. Okay, so let's move ahead. And let's see the next question pretty quickly. And the question is, outbreeding is an important strategy of animal husbandry because it helps in accumulation of superior gene. Remember, I am talking about outbreeding, is useful in producing pure lines of animals, is useful in overcoming inbreeding depression, or exposes harmful recessive genes that are eliminated by selection. So, why do we do outbreeding? What is the definite advantage of the same? Can any one of you tell me what could be the definite advantage of the same? All right. Here, the correct answer is option C. So, it prevents inbreeding depression. So, in order to get out of this phenomena of inbreeding depression, outbreeding needs to be done after few generations of inbreeding, right? So, that is the correct option over here. And let's move ahead and let's see the next question of the day. And the next question is, to obtain virus free healthy plants from a diseased one by tissue culture technique, which parts or parts which part or parts of the diseased plant should be taken? Is it the apical meristem? Is it the palisade parenchyma? Is it both apical and axillary meristem, or is it epidermis only? So which of the plant parts are you going to take to ensure that there is no virus in the new plantlets which you are obtaining through the technique of tissue culture? Right. And I guess the correct answer over here is, somehow the chat is not working. I don't know why. And the correct answer over here is option C. Both apical and axillary meristem cells are devoid of the viral infection. So they can be picked up for my culture, right? For plant tissue culture and for culturing and growing new plants in vitro. Okay, let's see the next one. This is another easy and an interesting question. So, consider the following four statements 1 to 4 and select the option which includes all the correct ones only. And single cell spirulina can produce large quantities of food rich in protein, minerals, and vitamins, etc. Is this statement true or false? Let's figure that out. And of course, it's a very true statement. Right. And then we have the next statement. Okay, just a second. So then we have the next statement. Body weight wise, in the microorganism, Methylophilus methylotropus may be able to produce several times more protein than the cows per day. So this option is also true. So my correct answer should include definitely these two options. Common button mushrooms are not a rich source of vitamin C. This option is false. Whereas a rich variety has been developed, which is very rich in a rice variety has been developed, which is very rich in calcium. This statement is also false. So over here, option D is correct. Just a second, everyone. Kindly hang on. So I will be available to all of you in another few seconds. Just hang on. All right, everyone. So I'm pretty much here, and let us just see what is happening. And I hope things will work fine for all of us now. Just a quick moment. Okay. All right. So I'm here again. And let's hope the chat will work now for all of us. Oh, well, well, well. Now I can see the chat is working. That's great. Yes, so it's working now. And thank you, Lovepreet Kaur. And let's move ahead. So it's a very interesting session which is going on here. We are quickly revising the questions and their reply. So I hope you all are at least watching the stuff, if not able to chat. Finally, you guys are able to chat. That is pretty good. And the next question is Hemgiri developed by hybridization and selection for disease resistance against well. Looks like everyone knows the answer. And Nikhil Swapnil, the correct answer is option D, definitely. Right. It's a variety of wheat which is resistant to rust pathogens. Right. Hello, everyone. Hello, welcome back. So, it's a variety of wheat resistant to rust pathogens. And let's move ahead. Okay. So, mutations can be induced with IAA, ethylene, gamma radiations, or infrared radiations. So, what is going to be your answer over here? So yes, everyone is pretty smart here. Akash says it is option C. Sudarshan says it is option C. Smriti says it is option C. Meghna, it cannot be option B. Option B is a gaseous hormone for plants. It helps in the early ripening of the fruits, right? And early senescence of the plant parts. So over here, the correct option is option C, gamma radiation, right? So that is the correct option which you have to choose because gamma radiation will directly pass through the cell. It will affect my DNA, it will alter its structure, and is a basis for causing mutation. So it is a big mutagen, one of our very potent mutagens in our world, right? So if anyone is exposed to gamma radiation, surely IAA is indole acetic acid, which is a kind of auxin. Yes, it's a growth hormone. Right. So compared to a bull, yes, Suhas, you are right. So compared to a bullock is docile because of higher levels of cortisol, lower levels of blood testosterone, lower levels of adrenaline, noradrenaline in its blood, higher levels of thyroxine. So Abreen says it is option B. Okay, what about Meghna? What about Aditya? Pavan says it is option B. Hasana says it is option B. Akash, Smriti, Aisha, Sushma, Sudarshan, Lovepreet, all are telling me it's option B. And yes, I'll go with all of you. So you all have your basics pretty clear. Bhanu, it is option B this in this case. Okay, here is an interesting one which will test your memory. So triticale, the first man-made cereal crop, has been obtained by crossing wheat with, is it rye, is it pearl millet, is it sugarcane, or is it barley? Okay. So Nikhil says it is option A, Smriti says it is option A. Yes, every one of you is correct. It is definitely option A in this case. Love Preet, it is option A, dear. So triticale is a hybrid plant obtained by the crossing over between a wheat and a rice variety. All right. And the next one. The name of Norman Borlaug is associated with, I guess all of you must be knowing this. No one should get it wrong. Okay. Yes, yes, you all know it. Right. Sandhya, it has to be option A, Green Revolution. And Norman Borlaug worked with one Indian scientist, Mr. M.S. Swaminathan, Dr. M.S. Swaminathan. We all know about it, right? To usher the great Green Revolution in the Indian scenario, right? Yes. So Norman Borlaug tied up with M.S. Swaminathan, tied up with Norman Borlaug to usher the Great Green Revolution in our Indian context. Okay. So let's move ahead and let's see the next one. India's wheat yield revolution in the 1960s was possible primarily due to what? Hybrid seeds, increased chlorophyll content, mutations resulting in plant height reduction, quantitative trait mutation. All right. So, which will be the correct option over here? Well, not so much due to the hybrid seeds, but due to the development of plant varieties, especially varieties of wheat and rice, where the plant height is reduced. As a result, the crop loss due to weather vagaries, due to wind, due to rain, due to hailstorm was reduced. The crop was more resilient, right? It produced more amount of grains, right? And it could quickly grow up. So option C is the correct answer over here. So that is a slight tweak here. So option C is the correct answer over here, and I hope it is clear to all of us. What do I mean by quantitative trait mutation? That's fine, Abreen. That's fine. What do I mean by quantitative trait mutation? Can anyone explain that? Well, it simply means we are not increasing the quality, but we are increasing the quantitative output of the crop. Well, Sudarshan, that's the trick. You have to choose a more suitable option. Okay, let's move ahead and let's see the next one. So, what is the best pH of the soil for cultivation of plants? Well, this is a very simple one. If you can get it right, then it's beautiful. And the correct answer is, can any one of you just guess it for me quickly? Well, not option B, Nikhil and Lavi. Aditya, you got it wrong this time, dear. Yes, Lovepreet is the first one to give me the right answer. Option C. So most of the crops grow pretty well in a soil with a pH range between 4.5 to 8.5. Right, neither below it nor above it. Anjali Desai, the correct option was option C. And this is another simple and an easy question. Honey is what? Is it acidic in nature? Is it neutral, alkaline, or basic after some days? So this is another easy one, not a very difficult one. Okay. Umbreen says it is option A. What about others? Lavi says option B. Nikhil says option A. Okay, well. Meghna, you are absolutely right. Nutan, the correct answer is option A. Honey is acidic, surely. Slightly acidic. Yes, Anjali, option A is correct. Okay, let's move on to the next one. And this question talks about the very process of pasteurization of milk, which involves heating milk for how much time at what temperature. So that is our question. Is it 60 minutes at 90 degrees Celsius, 30 minutes at 50 degrees Celsius, 30 minutes at 65 degrees Celsius? So, or 60 minutes at 100 degrees Celsius? Well, the correct answer is option C. So no one could get it correct for me. Okay. Well, the answer is this. Just check out the whole process of pasteurization. Right? Slight heating above 50 degrees Celsius of the milk and then immediately cooling it, giving a big temperature shock to all the bacteria in it and thus leading it to its demise, right? Okay, let's move ahead and let's see the next one. Yes, option C is correct. Okay, haploid plants are preferred over diploids for mutation studies. Why is it so? Is it recessive mutations express immediately, or induction of mutation is easier, culturing is easier, dominant mutation expresses immediately? Yes, this way you surprise bacteria. Okay, Aditya says it is option A. Yes, logically, if you all see, if you have two sets of chromosomes, right, there can be one dominant allele, one recessive allele. You might be trying to alter one or both, right? So, first and foremost, you have to induce simultaneous mutations in both the alleles. So, that becomes a problem, right? One may be different from the other, and thus the expression may vary. So, it's better to have a haploid set of genes, right? Or a haploid set of chromosomes. And whatever mutation is induced into it will be immediately expressed, right? So the option A is the correct option over here. Okay, let's move on to the next question. So, which of the following is correctly matched for the product produced by them? Well, this seems to be pretty interesting, and this is all memory game, right? So we are finally entering into the domain of microbes and human welfare. So which will be the correct option over here? Akansha is the first one to give me the correct reply. Yes, we all know this, right? Baker's yeast or brewer's yeast, Saccharomyces cerevisiae, is leading to the production of ethanol. Yes, you all know it. Yes, the very process of fermentation. Okay, let's see the next one. That was pretty easy. Which of the following is wrongly matched in the given table? So, can any one of you guess this for me? Which out of the four has been wrongly matched? Let's make it quick. Okay. Well, Sisher, Akanksha, Sandhya, Sanjana, my God, Rashi, the option which is wrongly matched in this case is definitely option C. Clostridium botulicum does not lead to the production of lipase. In fact, yes, yes. So therefore, this says the wrong option or the wrong matching. Let's see the next one. And the next one is pretty easy. Match column 1 with column 2 and select the correct option using the codes given below. So, citric acid is produced by what? It is produced by aspergillus. Cyclosporin is produced by what? Well, this is all memory game. Produced by trichoderma. Statins are produced by definitely monascus. And butyric acid is produced by Clostridium. Yes. So the correct answer is option B here. I hope all of you know this now. Scroll the page. Okay. I have already scrolled it quite a lot. And let's see the next question. And the next question is this. The next question is this. And I have again scrolled the page a bit so that we can see all the options which are there in front of us. Match the following list of microbes and their importance. So, which is the correct option over here? Okay, someone is telling me. Okay, everyone is giving me all sort of different answers. Well, Saccharomyces cerevisiae leads to the production of ethanol, right? What about Monascus purpurus? Purpurus. This helps in the production of blood cholesterol lowering agents, right? What about this one, the next one? Trichoderma. Yes, yes. The correct option is option A. Right. You all know it. So the correct option is option A this time. Okay, let's see the next one. So, this is all memory game. That's why I have kept so many questions of the same sort so that we can actually remember this stuff, right? These are some of the few questions which are going to appear in our examination. And here, Monascus purpurus is a yeast used commercially in the production of. I think we have been doing it for quite some time now. And yes, Smriti, the correct answer is option D. So the power of D came to our rescue this time. And let's see the next question. So, which one of the following is not a biofertilizer? So, is rhizobium a biofertilizer? Nostoc a biofertilizer? A mycorrhiza a biofertilizer? What about agrobacterium? Okay, the correct answer, science and tech, is option D. Remember, agrobacterium is used as a mediator of transformation in plants. Right? We have learned about agrobacterium mediated transformation in our biotechnology chapter. So, option D is the correct option over here. The rest three are definitely my biofertilizer. Rhizobium acts as a biofertilizer when it goes in for a symbiotic association with plants. Nostoc is a free living blue green algae. Mycorrhiza is a symbiotic association of fungi with plant roots, right? Okay, let's move ahead. Organisms called methanogens are most abundant where? In a cattle yard, in a polluted stream, in hot springs, or in sulfur rocks? Yes, yes, you all are correct. It is option A. In a cattle yard, methanogens, right? Right. Even the fat of the cattle contains a lot of methane gas. Right. And that's why the cattle dung is an ideal thing for the production of biogas in a biogas plant or a gobar gas plant. Right. So that's a very simple one. And let's see the next one. Which of the following is not used as a biopesticide? Can any one of us answer this? So we have got many in front of us, many options. Which of the following is not a biopesticide? Okay, so many people are saying it is option C. All right, some people are saying it's option D. Well, the correct answer is option D. So, this is not a biopesticide. This says going to harm my plants. So, the correct option is option D here. Okay, what is true about Bt toxin, right, which is obtained from Bacillus thuringiensis? The inactive protoxin gets converted into active form in the insect gut. Bt protein exists as active toxin in the bacillus. Okay. The activated toxin enters the ovaries of the pest to sterilize it and thus prevents its multiplication. Well, a wonderful theory. The concerned bacillus has antitoxins. Well, the correct answer you all know. The power of A. Option A. Sanjana, it's option A. So let's see the next one. Which one of the following proved effective for biological control of the nematode disease in plants? Well, this is a wonderful question. And let's see what's the answer for this one. Yes, finally a tough one for all of us. And the correct answer is option D, guys. So, this is generally used for the control of nematode disease in plants. Option D is the right one over there. And Let's see the next question. Let's make it quick. So, which one of the following is being utilized as a source of biodiesel in the Indian countryside? This is a very important question, even from general knowledge point of view. Right? So, biodiesel is becoming a reality of our Indian um, fossil fuel industry. And the correct answer, I have already boxed it. It is option A. Good evening, Jyoti. Well, the answer is option A this time. Okay, let's see the next one quickly. An aquatic fern, which is an excellent biofertilizer, is. Let's focus here, Srivastav Mridul. Is it Azola? Is it Pteridium, Sylvania, or Marsilia? Yes, Akanksha, you are right. The correct answer is option A. We are making it quick. So, which of the following is non symbiotic biofertilizer? Is it VAM? Is it Azotobacter? Is it Anabina? Is it Rhizobium? Okay. And the correct answer is option C. Anabina is a blue green algae and it is a free living organism. It's a prokaryote, right? Even without any symbiotic association, it can go in for nitrogen fixation. So it is a non symbiotic biofertilizer. Okay, one of the major difficulties in the biological control of insect pest is what? Let's make it quick. What's van? Well, it was VAM, right? And that is an Italian name for the mycorrhiza over there. It is actually a mycorrhiza. And here, Akanksha has given me the right answer. The predator develops a preference to other diets than the intended pest, which I am trying to clear off, or it may itself become a pest. Right? So that is the right answer. Okay, let's see. Okay, that is it for the day, boys and girls. We are done, and we have a link in the description. You all can fill in your details and get some help for preparation in your NEET examinations. And that is it for today. See you all again this coming Friday at 3:45 p.m. Indian Standard Time. Till then, take care, do good, and all the best. Bye bye, everyone.